Hello, my name is Laide and welcome to my series of presentations on CAR T-cell therapies for my case study, Pharm001B. So as I said, this case study is going to be providing an overview of all the life cycle activities for a CAR T-cell therapy, and it's going to be from product development, preclinical studies through to clinical trials, marketing authorization and post-approval. So it's quite a big, ambitious study. However, I have all the time in the world to do it, since this is the most that my career is going to go in terms of science. So I might as well take my time and enjoy the process. So I just want to say that these are theoretical products. They're not actually real products that are in any clinical study or in marketed or anything like that, even in marketing authorization. These are just theoretical. And I've got this company, LAF Therapeutics Limited. It's a fictitious company, and I'm using it as a way of exploring all of the options and the tools that are available to a small to medium-sized enterprise that could be developing a product like this. So it's my project, and it's my little, and my little case study, well, quite a big case study, and I hope you enjoy it. So my quote is, "Innovation is the ability to see change as an opportunity and not a threat." So I don't think there is a specific author for that quote. And this is particularly relevant and applicable to me, I would say, and I think to other people as well. And that is that, that we should take advantage of change when it's there, and the opportunities that we are presented with, and that we should understand that it's not a threat. Technology is not a threat, and the ability to make things transparent to other people so that they have access to information and they have access to knowledge is not a threat. And I think that it's wonderful to have the transparency and access to knowledge and information, and to expand my knowledge on this subject area. And I really enjoyed this process. So before we dive into the second part, because this is part 2 of my case study, I just want to give you just this overview again, because I've already provided it to you in my first presentation. So I'll just quickly run through it. So this overview just shows the life cycle activities that I'm going to have for my 2 programs. My 2 programs are LAT101 and LAT202, and they're 2 CAR T-cell therapies. One of them is autologous and the other one is allogeneic. And so this is what my case study will comprise. It will comprise of taking these products from conception through to development, the non-clinical studies that are required for clinical trials, then your marketing authorization applications or your biological license applications. In your clinical trial section, we'll be doing IND, investigational new drug applications as well. And marketing authorization will include MAA applications and biological license applications as well. We will also be talking about value demonstrations, so what the health technology assessments would look like, and we'll be looking at post-approval strategies. So today, and this presentation is going to be focused on Milestone 1, because I still have some things that I wanted to talk about, and particularly specifically the target product profile, because this is going to be a core development tool that I'm going to be using and updating. It will be a living document, so I do want to talk about that. And I also just want to go through some of the guidance documents and the information that we have available, and what that tells us. Right, so I'm going to be going back to first principles again, and I just want to talk about the LAT 101 because I've sort of struggled a lot to get to where I am and to get to this particular sort of construct, or at least I have had to sort of, learn a lesson or two. But this is what I think it's gonna be. So this is gonna be a tandem CAR T cell therapy. So it's going to be bispecific, and the two antigen recognition domains are going to be CD19 and CD22. The activation signal will come from the CD3 zeta, and the co-stimulatory domain will be CD28. The transmembrane domain will also be CD28, as will be the linkers and spacers. So for this particular product, it will be delivered via a lentiviral vector. So the reason being is that this will be inserted into the host cell genome, as you know, and that will provide a durable and stable expression. And then that's the main reason. And I'd also wanted to make the point that this is going to be a single gene approach, which is much more simpler than this multi-gene approach, and it's likely to work. I can't see the multi-gene approach working for this chimeric antigen receptor type molecule. So really, what I mean by that is that everything will be. There will be one single transgene that will translate the whole of the protein in one go. And so you get all of the components, the CD3 zeta, the co-stimulatory domain, the transmembrane domains, and the antigen recognition domains, and all the parts that are required to construct this CAR T cell, this CAR T receptor, T cell receptor, will be delivered in the one single chain, or the one single CAR construct. And then all the components are going to be fused together, but it will be one long polypeptide chain that's encoded by this single gene. And I feel that the lentiviral vector is the best approach because obviously it can transfect dividing and non-dividing cells. It's also has the payload, the vector payload, that can, allow this durable expression. There are going to be modifications that are going to be made to prevent the risk of replication competent viruses and the generation of replication competent virus generation. And obviously there are risks with regards to insertional mutagenesis. So we have to be mindful of the viral, is it viral or vector copy number. So all of those things we'll discuss later. Anything that goes into human beings have to be manufactured to GMP everywhere worldwide, as far as I'm aware. So this will be manufactured to GMP. And for the manufacture of the vectors and the master cell bank and working cell bank, you'll require a separate CTT dossier when you're making your submission applications. I think that's... Also, I think I've discussed this in other presentations, but your vector is going to be designed or modified to prevent certain risks. I've talked about replication competent viruses, but also and insertional mutagenesis. But the modification of these self-inactivating long-terminal repeats will also be included in this this design. And here again, this is the LAT 202, the second program and the second product, and this consists of two separate CARs. So one CAR is targeting MUC1, MUC1 is the aberrant high glycosylated MUC1, and the other CAR will be targeting HER2, the binding domain or the antigen recognition domain. This is going to be manufactured via the genome editing. So this will be it requires induced pluripotent stem cells manufacturing protocols, because obviously there's going to be gene editing and there's going to be knocking out of genes and knocking in of other genes as well. This is just this particular I would call it cartoon representation, is just displaying the chimeric antigen receptors. It's not actually displaying the synthetic receptors that are going to be present for this LAT202, because there are going to be two other synthetic receptors that are going to be there. But we've got the activation signals that's coming from the CD3 zeta, which is extracted from the CD3 complex, in the same way as LAT101 CD3 complex that really in the endogenous or T cells you have the CD3 complex, and CD3 zeta is the part that's the inside of the cytoplasm. It's the internal domain, and then you've got this co-stimulatory domain. So it's all linked together, and it's all constructed so that everything is in the one construct, if you like, and it's not, you don't have a separate receptor, CD28 receptor, as you would in the normal state, normal situations for your normal endogenous T cells. So this is the LAT202. So it's a bispecific, two different CARs, two different chimeric antigen receptors, as illustrated there. It's targeted towards two different antigens, and this is an AND gate. So this requires both MUC1 and HER2 binding, or recognition and binding together in order to activate those T cells. So it's an AND gate, and we have designed it in this way in order to increase specificity and reduce on target off tumor toxicity. So this is the second program that I've designed. I say we, it's me. I've designed this, and this is my proposal. Oh, and this is just a product description slide, which I think I've been through before. It's just LAT101 is an autologous CAR T cell therapy generated by ex vivo transduction. So this is the lentiviral route, yeah? And it's designed as an OR-gate tandem CAR targeting CD19 and CD22. So what that means is that the T cells will activate if you get antigen recognition and binding with either CD19 and CD22. The product is engineered with a co-stimulatory domain CD28 and includes an inducible caspase 9 safety switch introduced by gene editing technology, and that will probably be the CRISPR-Cas9 technology that is introduced. I still need to have a look into that, so this may change. I may decide to another means of introducing that inducible caspase 9, but for the time being the concept is to go via this gene editing approach, but I think there can be difficulties with off-target editing, and potentially for T cells. So we may have to think about that, but that's something for another day later on in the in the development, in the as I explore and as this project evolves. LAT101 is a bispecific CAR T cell therapy, I should have there, designed to activate T cell upon recognition of either antigen, thereby reducing the risk of antigen escape. And this is the reason why we do this, because sometimes when we in products where we have targeted CD19, if CD19 becomes underexpressed, then you get this cancer escape. So it's able to escape our product. And therefore, if you have two targets, then hopefully you can capture that, all of the B cells, because it's this product is intended for the treatment of relapsed or refractory B cell malignancies, including diffuse large B cell lymphoma and acute lymphoblastic leukemia. So this is the first one, and this is a product description for the first one. And here is the second product description. So this is LAT202, and it's an allogeneic CAR T cell therapy, CRISPR-Cas9 engineered, and it's to implement an AND gate bispecific targeting mechanism against aberrantly glycosylated MUC1 and HER2. So the AND gate means that both of the MUC1 and HER2 must be activated, or they must be binding, or antigen recognition and binding must occur for that T cell to be activated. And you probably have more than one of that, because, as I said, the T cells are peppered with or covered with these receptors. It's not just the one receptor. The product, as I said, the product comprises of two distinct CARs, each with one co-stimulatory domain, CD28 on the MUC1 targeting CAR and 401BB on the HER2 targeting CAR. And LAT202 T cells are further CRISPR-Cas engineered to resist TGF-beta mediated immunosuppression. So the TGF-beta immunosuppression, TGF-beta ligand is really there as an, what's it called? Homeostasis. Is that not homeostasis? I think it's immune homeostasis molecule, cytokine actually. And ordinarily, it's there to turn everything down once your immune system becomes activated, either as a result of viral infection or cancer or tumour. And once that's been cleared up, you want to bring everything down. You don't want the immune system to be continually activated, or else you might get autoimmunity, or you might get issues with that. And so this TGF beta, in a normal sense, brings everything down to a balance. Yeah. But what happens is that tumor cells specifically, they over-secrete and they overexpress and they over-secrete this TGF-beta. And what happens then is that when the TGF-beta binds to your normal T cells, you get the inhibitory response, and basically the T cells just are no longer able to become activated. So it's a way of the tumor cells evading the immune system. And so what we have done in this particular example is that we have flipped it. So the TGF-beta receptor that's going to be on the T cells have been either bound to either CD3 zetas. If it's bound to CD3 zeta, then it's going to be activating. If it's bound to the CD28, because I haven't decided which one, it would be co-stimulatory. And so therefore, what it isn't inhibitory. And so as a result of that, it means that that you can now, in the presence of this tumor microenvironment, instead of the T cells being switched off, they're activated and they're able to target the cancer and to kill the cancer or the tumor. And so that's the reason for this engineering, to resist the TGF-beta mediated immunosuppression. And then we've got this additional, we've got the inducible caspase 9 safety switch and the expression of this chemokine receptor CXCR2. And this CXCR2 is basically there for trafficking, so it guides the sorry about that, my mouse and my whole thing is very, very sensitive. I don't know how to stop that. But CXCR2 is a receptor, and when it binds to a number of ligands, it can then be guided towards from a low concentration to a high concentration, and you can use it to guide the T cells to the tumours. Right, so the next part of my is the target product profile. I just thought I'd just have this introductory slide. I just want to have a quick talk before I lunge into it, because I didn't have a lot of space on the slides to put a title. So this target product profile is a strategic development planning tool, yeah, and what it does is it's a blueprint for development. It's what we use to develop our product, to guide us with our clinical trials and our regulatory commercial planning throughout the whole of the product life cycle. So it's kind of like a vision document for the final marketed product, and it's got a whole host of information in there which I'll go through, but it is a living document. This information, I'm going to include it in my life cycle management manual when I get around to doing it. Kind of have an idea how I am going to do it, and so it will be included. It's an important and an integral part of that book, or it will be a manual. Oh, so this is the target product profile for LAT101. I don't know why I did this. There's nothing wrong with it. Yeah. So this is it. I did that because I didn't have enough space, basically. So LAT101 is the code that I've given it, and I think LAT101 comes from LAF therapy, LAF Therapeutics Limited, which is the fictitious company. So I've just given it a number following it, and it's just the unique company identifier for the code, for the product code, and the program, I guess. This product is an ATMP. It's an advanced therapy medicinal product, and that is defined by the ATMP regulation and the directive 2001/83/EC. Yeah, so it's a cell-based medicinal product, and the reason why it's cell-based, even though it's genetically modified, is that it's based upon its mechanism of action, its functional activity, and how does it behave in the body on a cellular level. And on a cellular level, basically the T cells bind with the target antigen, and on binding they become activated, and then they will kill the cells by apoptosis. So it's a controlled cell death. And so that action, even though the T cell have been designed by genetic manipulation, yeah, even though that's what's occurred to make that happen in vivo, on a cellular level, or molecular or cellular level we're talking about here, the interaction is because of the fact that the binding is what causes the T cell to kill the cancer or tumor cells. So that's what makes it a cell-based medicinal product. In the US, however, this is considered to be a gene therapy medicinal product, and it comes under section 351. Is it 351? Yeah, 351 for biological products, and that's the reference for the title Code of Federal Regulation, parts 312 and 600 to 800. But basically, it's a gene therapy medicine in the United States because of the fact that it occurred the whole concept is as a result of genetic manipulation. So both regions have a different approach with regards to how they classify the same kind of product. The indication, we've already been through that. The patient population is for adults and pediatric patients with CD19 plus or CD22 plus B cell cancer. So we'll do a test to make sure that they do have that CD19 plus and CD22 plus B cell. And specifically, we're looking at high risk groups, because we want to if groups that are not high risk, there will be other options for them. This is a product that is still in its development phase, and we want to make sure that we give it to patients who have very little option, and therefore we reduce there's a better benefit and the risk is lower for them. But for other patients where there are other options, we know the safety profile and the therapy profile for those other optional drugs, and therefore it doesn't make sense to give them a product that is still in development and still where we don't know what the long-term effects and consequences would be for them using those particular drugs. Whereas if we have somebody who only has maybe less than a year to live, maybe, then it makes more sense for them to have this drug when perhaps there might not be any other type of drug. So that's the reason why we're going for high risk and high need groups. The mechanism of action, I've kind of discussed that, where you have antigen recognition, antigen binding, T-cell activation, proliferation, and this against the malignant B cells. The dosing, for the time being, the administration will be via IV infusion after lymphodepletion, and this is the standard current practice for CAR T-cell therapies. The dose, this is based upon existing CAR T-cell titration programs, but it may very well change. Safety profile, well, we understand what the impacts of cytokine storms are and neurotoxicity, and what we have in place with regards to the inducible caspase 9 switch. Contraindications, we've highlighted there, sensitivity and acute severe infections. The monitoring, I've just got this basically from what's already out there. Storage and handling, this is currently based upon what's already out there. Cryopreserved cells, obviously, because you want to be the logistics of handling and managing. Although there are not risks associated with using cryopreserved cells or cryopreserved agents, etc., and giving it to very terminally sick individuals, but you balance the risk against the benefits. Manufacturing platform, already discussed, lentiviral vector, closed system manufacturing to ensure that you get risk of aseptic manufacture. Differentiation, well, this is a dual antigen targeting and the prior platform data, so the reducing complexities with regards to assessments, and hopefully we can it can be there for patients where there is no current therapies because they've had relapse or refractory. Regulatory pathways, for the time being, we're starting with the UK, EU, US, and using all of the tools and standard procedures that are available for gene therapy and cell therapy medicinal products. The objective is for early engagement and continuous engagement throughout the development, and the efficacy endpoints. I don't know whether these are standard. They're standard endpoints aligned with existing marketing authorization and biological license applications. So these are industry standards. And the second LAT 202. So here, as I've already discussed, the patient population is HER2 plus or MUC1 plus advanced solid tumours. For the same reasons, we'll be choosing high risk groups where there are limited options. I didn't put the classification. Maybe I put it at the end of the last thing. But anyway, the mechanism of action, it's going to be dual specific, and it's going to be allogeneic. But that's not the mechanism of action. The mechanism of action is this dual specific, where the antigen recognition targets the HER2 plus and the MUC1 plus at the same time for that precision. CD28 and 401BB are the co-stimulatory domains. You've also got this TGF-beta resistance. So this is the immunosuppression effects of the tumour microenvironment. So we've got something to counter that. The IC9 safety switch. Again, it's in most clinical trials at the moment, and the CXCR2 for tumour trafficking. So this is what differentiates this particular product from others. Dosing, single IV infusion and repeat dosing is possible because of the fact that this is an allogeneic. So it's not the same autologous system. It's not the same kinds of cells. So you're going to get it is possible, and also the advantage with allogeneic is that they can get the drugs quickly. So as soon as they've been identified, you don't have you go through the lymphodepletion, and then you can provide the drugs. You don't have that vein to vein wait time. The dose regimen, this is what I've got for the time being, but it may be adjusted, and it's based upon what's currently out there, and the efficacy endpoint. Again, ORR, CR, so the overall response rate, complete response, progression-free survival, and the overall survival. And these are reflected in your standard clinical trial applications. I don't know what else we've got here. I don't think there's anything more that compared to what I've already stated. It's the same kind of classification, but these are going to be these target product profiles are going to be our starting point. They were living documents, so when they'll be version controls. So when they're updated, it will be a new version number, so that we can look and track and audit the history and see what the changes were, justify the changes. But basically, the only difference here, I guess, is the manufacturing platform, and that's this allogeneic. So you'll be using gene editing for immunogenicity. So we'll be knocking out the HLA, the relevant HLA genes, so that we don't get that graft versus host disease, that you don't get that rejection. And also, we'll also be knocking out the T-cell receptors as well. So you don't get the graft versus host diseases from the T-cell receptors. So there'll be a number of editing. So there are certain risks associated with this. I don't think I put that on there. You can't, if you're not careful with these target product profiles, they can be very long, I think. But there are going to be certain risks associated with gene editing, and that will be things like off-targeting, off-target editing. And there are also unknown risks that we are not yet aware of relating to on-targeting editing. So there are certain risks associated with this that are unknown, and that's why we go for a certain patient population. So I just had some considerations for LAF Therapeutics Limited. So LAF Therapeutics Limited is this fictitious company, and if we're going to look at this as a small and medium-sized enterprises, what are the things that we're going to be thinking about? So these drugs are for subjects where there's an urgent need. It's life-extending drugs for patients with limited options. So the vein-to-vein time should be as minimal as possible. So particularly in cases of aggressive forms of cancer or tumour, and therefore the manufacture and release procedures should be as efficient as possible. Sometimes you have a patient that's accepted into a clinical trial, and then by the time you've manufactured a drug, they're no longer eligible for the drug. So you've got to really have some consideration with regards to your clinical trial data requirements and compassionate use, maybe. And also you have to make sure your manufacturing process is as efficient as possible. So things like testing, you might want to be using certain kits, especially microbiological testing, micro-testing. Some of them can take up to 2 weeks. So, and you may want to give the patient, or the doctor, the medic might decide they want to give the drug to the patient. They can't wait for the testing. So you can use, say, for example, these kits, mycoplasma kits, to perform your testing, and then in the interim, and the patient can get the drug. So there's just something to consider. Another thing from that, these are just notes that I was writing down. So, there's a lot more to it than this. There's just some notes. And that's to do with I don't think there'll be transduction efficiency. I think I was reading somewhere that with regards to tandem cars, that you might get issues with transduction efficiency. So we should just maybe monitor that. But the cargo is not that, it's not that significant. It's well within the 8 kilobases. So I think it should be okay. But I think I've just got that as a note. With regards to the binding domain, so there's a potential for, if we're using murine sources as a binding domain, I don't know whether that's still the case now, but if we are using murine sources, just to bear in mind that could be a potential for an immunogenic response. The patient's immune system might attack it. You may decide to fully humanize it, and if you do fully humanize it, you need to just check to see what effect, if any, that has on the ability of the antigen binding site to recognize the site and to bind and to produce that stable activation. And then again, variability in the starting material is going to be could be potentially a major source for problems, in the sense that, particularly with patients with aggressive forms, they may not necessarily be able to produce the white blood cells that will, or the leukapheresis samples that are maybe going to pass the initial criteria in order for manufacture. So you just have to bear that in mind. Yeah, and then this is as a result. So these notes are as a result of reading a guideline, and I'll point that guideline out to you because I've listed it at the bottom. Well, never mind. Just certain things to take into consideration. I think that this is what I've said with regards to your starting material, the patient's T cell populations. You may need to control that. And, you're going to have to look at things like, the number of viable cells, the cell count, the cellular composition, how much of what is there, how much T cells, how many B cells and natural killer cells, because that can have an impact on the quality, and also potential for contamination. So the starting material should really be tested for microbial contamination to prevent it from entering the manufacturing process. You can also get cancer and tumour cells in your original samples, and you have to sort of remove them in your final product, and you may not be able to necessarily remove them, so you might have to include something in your specification as to what you think is acceptable in terms of the benefit risk profile. Good tissue practice, the chain of custody, especially for autologous products, you do need to be able to state that this is the patient. This patient that is receiving the drug is the patient that was donated the original starting material for autologous products. So this chain of custody will also include, the chain of custody is just basically the documentation. You need to have sort of, numbering system, an alphanumeric numbering system that will account for, that will identify the actual donor and the chain of identity. And you also would include in there short tandem repeats, ID testing to confirm that this is genetic testing to confirm that the patient, the donor and the recipients are the same, as well as also checking that there isn't any contamination from any other recipient's material. And quality control and quality assurance. Your quality control is your release specifications and testing. Quality assurance is your whole process, your whole manufacturing or testing or collection process, etc. So these are just notes that I was making from a guidance document. I'll talk about that guidance document later on, and just these are the things that we need to consider, because there's a lot of guidance out there. There's a lot of support. There's a lot of processes and procedures that regulators provide to developers, so that we can have a product that is going to be successful, so that patients can get access to these medicines as quickly as possible, that we provide the data that's required, the correct data, the scientific data that's required in order to market these products, in order to allow these products to be registered in clinical trials and investigational new drug applications. So they want us to have this information, so there's no point in reinventing the wheel. It's already out there. The information is there for us to take advantage of. So this is some of the things that we have to consider. From a CMC perspective, the chemistry, manufacturing and controls, the cryopreservation agents contain DMSO, and that might be problematic for certain patients if they're hypersensitive to it. So you've got this patient reaction with the cryopreservation agents. I think that it makes sense to freeze, cryopreserve your final drug product. It makes it easier with regards to shipping, and logistics. It just makes it easier. There are going to be thawing processes and thawing procedures in the clinics, and that has to be standardized, and those procedures have to be trained. The medics and the relevant people who are going to be administered the drugs have to be trained, and you have to make sure that they do it in the correct manner, and if they don't, if they deviate, then that has to be what's the word I'm looking for, captured, and it has to be recorded and included in the clinical data that we are going to analyse as a result of clinical trials, or even as a result of marketing authorization or biological license application. So these are the sort of things that you need to be considering in use stability and compatibility. This again is the final drug and how it's used. We need to show that the preparation is not going to go off. It's not going to be no longer have its potency. It's not going to have its therapeutic benefits, and it's compatible with the delivery devices or the administration devices that we're going to be using, or the medic's going to be using in order to administer the drug to the patient. And your QC release specification, that's something and there are a number of tests that's going to be on there, and one of them is going to be the vector copy number as it's a requirement. And I was just thinking about the vector characterization. Again, this is information that's available in guidance documents, and the guidance documents I will be listing at the end of this presentation. And there are certain things that you have to have in place in terms of characterizing your vector. Identity, purity, potency, sterility, and mycoplasma. So identity, you need to be able to confirm your DNA sequence, and that comes from your source material, the source DNA plasmid that's used in your HEC293T packaging or producer cell line. So these cell lines, they're transfected with multiple DNA plasma. And that's how we manage to manufacture the viral particles in order to prevent recombination, and in order to prevent the potential for getting viruses, creating if you like new viruses. So one of the plasmids contains the CAR transgene flanked by your long terminal repeats, your self-inactivated long terminal repeats, and then it depends on how the manufacturing process is set up. And that's another presentation that we can discuss that, because there are various options like producer cell lines and packaging cell lines. There are various options and various ways, and in the quality part of this presentation I'll be talking about that, so I would have honed that down by that point. But your manufacturing process should be well characterized. You want to have a well characterized, consistent process for anything, really. And you really do also want to have a reasonably well characterized product as well, because in order to characterize your manufacturing process, you have to have some understanding of the characteristics of your product. You have to understand what the critical quality attributes are in order to understand how they're going to be impacted on by your manufacturing process, and therefore defining your critical process parameters. In your early phase development, the reason why you want to have a well characterized method, even if a manufacturing process, even if you're going to make changes to your manufacturing process, which, by the way, you should engage early with the regulators with regards to what your plans are, it just means that you've got that consistency, and it also means that regulators can make decisions, and they can make decisions on what questions that you may have for them. And it also means that your manufacturing process is the product itself. Making changes, significant changes that could potentially impact on the quality and thereby the safety and efficacy of your product at your later phase requires a lot more documentation and a lot more changes. The potential to do not just analytical testing, but also non-clinical and clinical testing. So identity, purity, potency. Identity to confirm purity, you're looking at impurities like host cell DNA, host cell proteins, residual plasmids. Potency and titer, you're looking at you're looking at your functional infectious viral particles. And that's going to lead you and give you information on your multiplicity of infection. Sterility and mycoplasma, these are standards. You need to do this. It's a critical safety test. And then comparability, because as I said, you're always going to be wanting to make changes. Start off as a small product, start off small, and then eventually it will grow, hopefully. And you want to be able to implement those changes, but in order to implement those changes, you need to do comparability testing. And comparability starts off really as analytical testing. That's how we primarily start it off, and we make our comparisons, and we're able to say that the product is similar. They're not going to necessarily be identical, but they're going to be similar in terms of quality, safety, efficacy, and a whole load of other things as well. And you can demonstrate this through analytical testing. Now, if you can't demonstrate it, if you can't detect what potential changes could be, then you may have to go back and do certain non-clinical and clinical studies. So that's the reason why you have to plan in advance, start with the end in mind. You have to discuss with the regulators once you've got honed down the manufacturing method and your product, and you have to discuss the kind of data that you would be required to provide if you made this change or that change in the future. You discuss that with the regulators now, then you can sort of see how to incorporate your changes, what changes you should incorporate earlier on in your development to reduce the risk of you actually having to produce more data. So that's the third point there, and that is that if the objective should be to make manufacturing changes in the early phases of development to minimize risk and burden of comparability studies in later clinical trials. Yes, and this is the non-clinical. So it's just certain considerations that we need to take into at this early stage. There are also clinical considerations which I didn't include in this presentation because I just wasn't. I think the clinical is going to be a whole big subject on itself. So I just thought I'd leave that. But these are just the early thoughts that are running through my mind as we are, going through this process. So this guidance that I will be showing you about later on, I don't know if I've got a screenshot of the actual guidance document. I don't think I have, but I have got it in my references and I'll point it out. It's basically giving us, or it's the FDA guidance, and it's about having a flexible approach for your non-clinical studies and understanding the unique safety profiles of on and off target specificity, particularly with gene editing and also with the lentiviral route, although we have enough information and experience with regards to that. But It's just about that case by case approach, because we understand that, not all of the CAR Ts in development, they're going to have different differentiators, and therefore there are going to be differences about them. You're going to be able to take one approach for one and not for the other. So it's a risk-based strategy. You want to have that early engagement. You want to decide, say for example, you want your non-clinical studies to model what would occur in humans. What is the human response? Does this give us an indication of as to the human response? Can we extrapolate the data? The animals that has been selected, are they suitable? Should we be taking the surrogate approach because it's more applicable, and it's more it's going to give us useful information on toxicology and pharmacology. So all of those things, it's basically a case by case approach and basis, and that they basically, the FDA have a flexible approach, and they will look at it on a case by case basis. So this is just it's a overview of the sort of things that we're starting to look at now. So this is the SME certification in the EU and OMS registration in the EU and in the UK, and the sort of interactions that we want to be making with the regulators via scientific advice, via ATMP certification, ATMP classification and certification, using the processes and procedures that are available for us, using the Innovation Task Force in the EU, ILAP in the UK, Interact as a means of having those early discussions. So these are just sort of, I think the message that I'm trying to make here is that this is about not just a regulatory engagement with the regulators, but early engagement with all of the relevant bodies, with the ethics review boards, with the HTA bodies, having that discussion as to what information they need, and providing and making sure that we get that information at the correct time, as opposed to having to go back and redo work or reassess data, and in order to get data that perhaps the HTA bodies require. So if we have that information sooner rather than later, we're in a better position to get their approval, if you like, so that these particular products can be marketable, and these particular products can be used by hospitals to the public. Oh, strategic foundations. Yeah. So I think for this particular product, it's quite clear what it is. We don't really need to do an ATMP classification. We don't need that. It's quite clear. I guess it's nice to have, and I think it's nice to get that confirmation, I guess, but it would just be like a rubber stamp. And I would have a discussion as to whether or not we should bother to get this classification with the regulators before I even applied for this particular product, because it's quite clearly a cell-based medicinal product. However, it's always good to have that tick box exercise where you've got that confirmation that no, we should treat this as a cell-based medicinal product. The reason why in the EU you want to have that is because of the way that the dossiers are going to be structured, and the way that the way that you present the information for authority to review. They want to see information presented to them in a certain format, in a certain order, in certain sections, if certain information should be in certain sections of the dossier. And that's the reason why, we want to know how to present the dossier, how to structure the information that's being presented. Yeah, these are just follow-on these are just follow-on notes that I made. And, this is about, I don't know whether it's possible you should get full characterization as soon as possible. I don't know full characterization, but a good well characterized manufacturing method is required in order for regulators to make their decisions with regards to ATMP classifications. But as I said, I think it's reasonably obvious what kind of a product this is. But it's just a general point. You want to have a well characterized manufacturing method. Anything that goes into human beings has to be manufactured to GMP, including anything that's treated as a starting material, and that goes for all modalities, not just for ATMPs. Your non-clinical programs, I think I talked about that. You have to use models that are predictive of human responses. Non-clinical programs are used, models and programs and studies are used to inform your clinical trials, safety of clinical trials, safety, efficacy, proof of concept, proof of principle and proof of concept. Before you conduct any non-clinical studies, and before we conduct any non-clinical studies, we should be going to get scientific advice just to make sure that we don't spend all that time, resource, doing testing in vivo testing, especially. In vitro testing just as much as well. In silico testing could be expense. There's an expense related to that, and time that's related to generating the data for that. Prior knowledge, where do we put some of that information, some of the literature sources, and also the fact that all of this is going to be captured in the life cycle management manual, which will be a living document that will be used to detail the development, regulatory and commercial roadmap of this product, of these programs. And then there's some other things as well. SME certification. So to be recognised by the European Medicines Agency, the LAF Therapeutics must have SME status, so you need to apply for that. That's also the case in the UK, I believe. Organisation Management Service registration. This is a mandatory requirement in the European Medicines Agency. You need to have it if you're going to be doing any kind of application. So the OMS is the EMA's master database for storing and managing data on organisations. So you have this unique identifier, and it's included, and it has to be included in all of your applications. And if I was going to do an ATMP classification, this is general for at the early stages of development. But as I say, I wouldn't imagine that we would need it for this type of product. But it's always good to just get that tick box thing that we don't need it. And also other countries may require it. It may be part of their requirements. So you might be wanting to go into other countries within the European Union, such as, say, for example, Switzerland. I do know that other countries can be quite dogmatic about information that they need. Do you know what I mean? Even though you think something is straightforward and clear, they may need it. So that's the reason why I might need to consider going for ATMP classification. And here are my references. Now, this is about, the first one is about MUC1 and glycosylation. This is about, I just had some issues with regards to whether or not it's a good binding site or good target. Chemistry, manufacturing and controls information for human gene therapy investigation on your drugs. I haven't gone through that in a lot of detail in this presentation, but it will be something that I will be covering. Human gene therapy products incorporating human genome editing. So this is an important document, I think. It talks about the specific issues and requirements for gene editing in addition to the CMC requirements. So I think it's a really important guidance document, and it's something that it's less than 2 years old, but I think it consolidates and puts all the information in one centralized FDA document, guidance document. ACell. This is a case study based approach and looking at the quality by design principles, basically, and that's how we would want to That's how we would want to approach this product, the manufacturing process. We want to build the quality into the manufacturing process. So it's going to be a quality by design approach. This is the TSC, not TSC, the tissue, good tissue practice guidelines to quality and safety of tissue and cells for human application. So that's the good tissue practice. This is the next one is the chemistry, manufacturing and control information for human gene therapy investigational. Oh, did I say that? I thought I'd already had that, but maybe not. This is about manufacturing of CAR T cells. So what the process parameters are with regards to, and this is with regards to qualification. So your vector manufacturing, and what the problems, not the problems, but the strategies are. And for CAR T cell in general, and then scalable manufacturing of CAR T cells for cancer immunotherapy. That's about comparability and looking at the scales and what we need to, just a review of the processes. So these are my what I have researched. Right. Guidance for industry phase 1. This is sort of good manufacturing practice. Basically, as I was saying, that in phase 1 clinical trials, you do have to manufacture your drugs to GMP. I don't know why anybody would think that you wouldn't, but there's the guidance that tells you that you should. Oh, I don't know if I've got that. I thought I had the guidance. I might tell you what it is later. And this was a YouTube video on lentiviral titration and viral titer, because you have to measure the amount of viral particles in order to get your multiplicity of infection. Please let me have put this. Oh, I didn't. So there was another guideline, and it relates to the considerations, and the FDA guideline relating to the considerations for CAR T cell therapy during development, and it was published in January 2024. So that was the guideline that I had reviewed, but I didn't include in my references. Many apologies for that. Or maybe I did and I just missed it. So I hope you've enjoyed my presentation. I've gone through it quite quickly. If you've got any questions, please don't hesitate to discuss them with me, or to leave a comment, and I'll try my best to respond to your comments. Thank you so much. Bye bye.